Welcome, everybody. Thank you, everyone, for joining today's CRB1 webinar with Foundation Fighting Blindness. Before we get started, I would like to briefly review a few details for this call. Currently, all participant lines are muted and without video. Please be aware that all controls are at the bottom of your Zoom window. This control bar may collapse when it is not in use. If you would prefer to prevent the controls from hiding automatically, you can use the following keyboard shortcuts to change your always show meeting control options. If you are using a Windows computer, you can press the Alt key, and if you are using a Mac keyboard, press Command and backslash at the same time. Today's presentation is being recorded and is available with closed captioning. To activate closed captioning, please select Show Captions at the bottom of the screen in your Zoom toolbar. We are so thrilled to have you today, and I would like to hand it off to Angela Bowman.
Wonderful. Thank you, Julie. Thanks so much, everyone, for being here. I know some of you have had a very long CRB1 intensive day because there was a CRB1 patient event in the UK. So for those of you who are joining us from that event, we welcome you. So I'm Angela Bowman. I'm the science advocacy lead at the Foundation Fighting Blindness. And I have spent considerable time over the past few years understanding the science behind inherited retinal diseases, including those caused by mutations in CRB1. Every year, the Foundation awards millions of dollars to support retinal disease research. One of the ways we support research is through a specific grant type called a Program Project Award, or as we abbreviate it, a PPA. PPAs are long awards, $2.5 million over 5 years, and are designed to help researchers tackle problems that are too large for a single lab to address. These awards require the participation of multiple researchers with distinct but complementary expertise, all focused on a single research hypothesis. Last year, we awarded a Program Project Award to Dr. Jeremy Kay at Duke University and his colleagues to better understand what drives retinal cell death in CRB1 associated retinal disease and to determine if we can stop or slow that cell loss. This project involves 8 labs at 4 universities working on 4 different but connected projects. Today, Dr. Kay and Dr. Alekseev, another member of the team, will explain what they hope to accomplish and provide an overview of CRB1 associated disease. We'll hear first from Dr. Kay and have a Q&A about his part, and then we'll hear from Dr. Alekseev with a second Q&A session. Please type your questions into the Q&A portion that you can find at the bottom of your screen. So without further ado, it's my pleasure to introduce Dr. Jeremy Kay. Dr. Kay is an associate professor of neurobiology and ophthalmology at Duke University. His research focuses on how cells communicate with each other during retinal development. His team provided evidence for different CRB1 isoforms or different flavors of CRB1 in the human retina, which led to new research directions for future CRB1 therapies. Dr. Kay will share an overview of the recently funded Program Project Award focused on understanding CRB1 associated retinal disease. Dr. Kay?
Well, thank you, Angela. Thank you to all the organizers for putting this together. It's really an honor to have the opportunity to tell you about what we've been doing in the lab and how excited we are for the project that we're just getting underway with FFB support. So I thought what I would do is just start by introducing the lab in general very briefly and kind of tell you the story about how we stumbled into our findings on CRB1. It's a real great example of how basic science and curiosity-driven science can lead in unexpected directions that have really big potential impact for understanding disease. So you don't always have to be starting with the premise that you're going to study the disease to still make great progress. And then I'll turn to telling you a little bit about what we're doing with the program project and why we're excited about this. Um, as Angela mentioned, I am in the neurobiology department as well as ophthalmology at Duke. I was trained as a neuroscientist. Since graduate school, I've been interested in how the brain develops and the visual system in particular. and sort of fundamental questions of how the brain and our sensory experience make us who we are. And from that very high level, I started becoming interested in how the nervous system is assembled during development to um, get us to the points that we all are at as adults. And so this took me down the path of being interested in um, proteins that are on the surface of cells during development, because that's how neurons and other cell types in the nervous system talk to each other and um, interact with each other in their environment as they're building the retina, other parts of the visual system, the whole brain. And so we were coming into this project, we were really just curious about what kinds of um, proteins are on the surface of cells in the developing retina to allow them to talk to each other as they're building the tissue. And it turns out that CRB1 is exactly the kind of gene that our lab typically studies. So, like most genes, like all genes really, CRB1 is the gene itself is a sequence of DNA, and that what that DNA does is it encodes the instructions for making proteins. And in this case, it's a protein that sits on the surface of cells. And the intermediary between the gene, the DNA, and the protein is RNA. And so, what we were doing, how we stumbled into this, is we were surveying the diversity of RNAs that make cell surface proteins in the developing retina. And we were surprised to find that when we looked at the sequence of the CRB1 RNA that we were seeing both in mouse and human retina, there was something funny going on. You can look up in a database what the sequence is supposed to be, because other people have put their information there from their previous studies, and ours was close, but it was not quite the same. And so, as Angela mentioned, this is a known thing. We call it isoform diversity. It means that the individual genes can make not just one RNA and one protein, but a few different RNAs and a few different proteins. And this is broadly true for many genes, but nobody had really uh, noticed this for CRB1 before. And the reason that people had missed this is that there's new technology just becoming available to better look at the isoform diversity of genes. And we were in the right place at the right time to use this technology. And what we found is that the most abundant RNA and protein isoform of CRB1 in both mouse and human retina was different than people had expected. It was not one that people had studied before. Um, and so we call the original isoform that people had been studying, we call it the A isoform for simplicity, and we call the new one that we found the B isoform. And so why was this discovery important? Um, I'm going to tell you first the big picture, and then I'm going to give you some specific examples of why this really makes a big impact for thinking about what causes CRB1 disease and how to treat it. So the big picture is that with knowledge of this new isoform, we are better able to understand. It's allowing us to do new research directions that will help us understand what the CRB1 gene normally does in the developing retina and how, when that goes wrong, and developing and mature retina, and how, when that goes wrong, um, this leads to retinal degeneration, to the photoreceptor cells, the light sensitive cells in the retina dying. Um, and if we understand what CRB1 normally does, this will give us ideas for how to create new therapies that will fix the problems that arise when it's not doing what it's supposed to do. And if those therapies, uh, if we get new ideas for therapies like this, it holds great potential to halt or slow degeneration and preserve sight. So that's the motivation for all of our research now in the program project. So here's some specific examples of what finding CRB1 has taught us. So first, the original isoform, the CRB1A isoform, had been studied for many years, and it was known to be produced by a particular cell type in the retina, which is not the photoreceptor cells. It's not the rods and cones that actually die in degenerative diseases. Instead, it's a supporting cell called a Mueller glia cell, a non-neuronal cell. So it was thought that the origin of the disease was with these non-neuronal cells. But when we found the CRB1B isoform, we learned that it actually is made by the cells that actually die in the disease. It's made by the photoreceptor cells. And so this insight really changed our thinking about which cells in the retina are involved in the pathology. And this is giving us new ideas about how we could intervene to make pathology improve and to save sight. Um, and in particular, our research has led us to the following idea, um, that the A isoform on the Mueller side and the B isoform on the photoreceptor side are involved in forming a very important and specific cell-cell connection. Um, it's a it's a junction, a tight connection between the photoreceptor cells and the supporting cells that's really structurally important in the retina. We call these connections collectively the external limiting membrane. Um, you can think of it as a belt of steel reinforcements that go through the center of the retina around the base of the photoreceptor cells that hold them in place, give them um, tensile strength that keeps it from folding or collapsing on itself. So um, it had long been thought that these junctions might be involved in the disease, but our discovery helped us come to a new idea, which is that you have one type of protein on one side and one type on the other side that are making uh, the key sites where CRB1 is acting to keep the retina healthy. Um, so another example um, is that the protein sequences of A and B, they're pretty similar, but not exactly the same. And from some of the differences that we found between the A and B, we've had new ideas about how the different cells that make the different isoforms might be using CRB1 at these junctions. And then the third concrete example is probably that will take a little more explanation. So, um, in order to understand at the kind of detailed level that would allow us to come up with new molecular therapies, um, why photoreceptor cells are dying in CRB1 disease, we need to be able to study this in animals. Um, but there wasn't really any example of an animal where if you made mutations in the CRB1 gene that you got photoreceptor death like you see in the human disease. And one of the reasons for this, it turned out we were able to show the key reason for this is because no one knew about the B isoform. So in human patients, I'm sure many of you are wondering, the vast majority of patients have mutations that affect both of these isoforms. So they're both important. But when people made the first mouse models, they didn't know about the B isoform. So what they made only affected the A isoform and not the B. And so those mice had very healthy retinas, and it was not possible to study why photoreceptor cells were dying because they weren't dying. So what we found is that if you make a mutation that better mimics what's going on in human patients where both isoforms are affected, now you get mice where the photoreceptor cells are dying. We call this a mouse disease model. The word model is a bit of a term of art because mice and humans, even though the mice are capturing key aspects of the human CRB1 disease, they're not exactly the same. So we use the word model to emphasize and remind ourselves that it's not exactly the same as what's happening in people. But we do know that it's capturing the photoreceptor death for the first time. So this gives us a tremendous opportunity to dig in and figure out why, when you lose CRB1, at least if you're a mouse, that the photoreceptor cells will die. Um, and so that brings us to the program project grant. So the motivation for this project was if we can understand why the photoreceptor cells are dying when the CRB1 gene has mutations in it, this will give us new ideas about therapies that can slow or prevent degeneration and thus preserve vision. And going into it, our work with our new mouse disease model led to the idea that defects at the external limiting membrane cell-cell junctions, the ones I was describing earlier, are the key underlying cause, or at least one of the key underlying causes of CRB1 disease. Um, and um, so the goal of this work overall, where we built a team, a multidisciplinary team from many universities around the country, all with the shared goal. And the goal is um, not just to continue using mice to test our hypothesis about why cells are dying, but also to see if this idea generalizes to other species, including humans. And so this is really important to establish the relevance of the mouse work that we've been doing and to uh, reassure not just us, but also federal regulators who might be looking at new therapies, that the mice can be sort of a way to establish the proof of principle that new therapies work. And then they would let us go on to testing new therapies in patients through clinical trials. Um, and then a key part of this as well is to see with our mice whether we can come up with any ideas for how to fix these cell-cell junction problems at the external limiting membrane where we think CRB1 is acting. And if we can fix those problems, do those slow or prevent degeneration in the mice? And if that were the case, that would give us uh, uh, leads on things that we could then potentially look at as human therapies. So the participants in this project are the participants in what they're doing are, so we obviously our lab, I've just told you what we're doing, but I'll tell you about the other folks. So-Hee Cho is a professor at Thomas Jefferson University in Philadelphia, and he's a real expert in cell-cell junctions and how proteins like CRB1 are important for building junctions like that. And his lab is using human retinal organoid model system. So they are making from stem cells from induced pluripotent stem cells. So they are able to, that means that they don't have to have actual human stem cells. They can take any old cell and turn it into a retina. It's an amazing technology. And they are using this to study whether some of the biochemical functions that we've been looking at in mice might pertain to the formation and stability of junctions in a human-like retina in a dish. Then Maureen McCall and Ron Gregg are professors at the University of Louisville, and they have expertise in developing pig models of human retinal degeneration diseases. And why this is really important is that mice have eyes that are pretty different from human eyes. But pig eyes are a lot more similar. So if we want to convince a company or federal regulators or people who could be gatekeepers towards taking our ideas from the lab into the clinic, that we have a good chance of all of this working, having a large animal model like a pig is going to be really important. So the goal of the McCall and Gregg lab work in this project is to take some of the ideas that we have developed in mice and see if they pertain in pigs. And so to do that, they're generating pigs with CRB1 mutations that are very similar to what are found in human patients, at least some patients. And then finally, there's the project that Dr. Alekseev here and our other Duke colleague, Sina Farsiu, are leading. So the goal of this project is to see if we can find evidence in human patients that the external limiting membrane is damaged early in the course of the disease. And the reason why this would be important is, again, it would validate that the mouse work is showing us fundamental biology that pertains to humans. And would inspire us to go further in looking at whether we can reverse these problems at the external limiting membrane in mice. It would encourage us to think that that could as well have a therapeutic impact in humans. Sina is a optical engineer, and he builds amazing devices to get unprecedented kinds of images of human eyes. And in collaboration with Dr. Alekseev here, who is a clinician who works with our CRB1 patient population at Duke, we are bringing um, uh, some of the patients who are early on in the course of the disease to Sina's lab to be imaged and look for evidence of damage at the external limiting membrane to see if what we're seeing in mice is replicated in humans. And then finally, what our lab is doing here is that we're testing the idea that a CRB1B based gene therapy could fix the damage at the external limiting membrane and halt degeneration. So the idea here is if we put so that normally the CRB1 gene is complicated with all these flavors and all these different isoforms, but what if we could simplify things and just use one of those and restore function of the gene? So that's the idea we've been testing. And in mice, what we found so far is that if we restore to photoreceptor cells in mice that normally don't have any CRB1 protein, if we restore the function just of the CRB1B isoform into photoreceptors, we don't completely fix all the degeneration, but it's way better. We don't completely fix all of the damage at the external limiting membrane, but it's way better. And most importantly, when we test these mice for visual function, for visual acuity, their acuity is indistinguishable from regular mice, which suggests that we might be on the trail of a therapy that could be really effective and potentially very exciting. So that's where we're all headed in the next few years with this grant. I'm excited to have the opportunity to tell you about it and answer any of your questions.
Okay, all right. Thank you so much, Dr. K, for that overview. Just a couple of questions. The first is what can cause overexpression of the CRB1 protein?
You mean in the disease context, or is this? So in the CRB1 disease, what normally happens is that we are losing expression of the protein. So most of the mutations, I think all of the known mutations are loss of function mutations. That is, if you dig into what they're changing in the genetic code, they're reducing the amount of protein that's made, or they're making the wrong version of it and it's degraded or it's just not made at all. There are some kinds of genetic diseases that are not quite this simple, but in the case of CRB1, it seems like the disease is always resulting from a loss of the protein. So then maybe the question was getting at how would you put it back? Um, what we've been doing in the lab is so far it's mouse genetic tricks that are not something you could do in a human patient, but we're starting some discussions with various folks in the biotech space about different ways to deliver the CRB1B back. Um, First, we would test it in mice, and then hopefully in the pigs, and then eventually in humans. So there's different options. That's an area of further research. In some cases, so there's approved gene therapies now for other diseases that use a virus to deliver it, but then there's also new non-viral ideas that may have some advantages. So that's definitely an area that still needs some work.
Okay, thank you. Another question that came in the chat is, did you find the treatment or care for retina MDG, please?
Retina MDG.
Correct.
I imagine that means macular degeneration.
Oh, well, that would be amazing. That's such a common one. Um, I have some really amazing colleagues at Duke who are working on that disease. Our lab has not been studying that disease because, as it turns out, the underlying biology is so different um, that our lab's expertise in cell surface proteins and how cells talk to each other wouldn't really be that helpful. But I can assure you that there's lots of folks who are working tirelessly on that disease.
Okay. Another question, what is the difference between isoform A and B? Are they CRB1 mutations in both isoforms or just one?
That's a great question. So the B is shorter, so it has a large chunk that's overlapping, but it's missing compared to A. It's missing some bits at the beginning, and then at the end. So this is all the part that's outside of the cell, and then the proteins are in the cell membrane, so they're kind of stuck on the surface of the cell. And then there's a part that's inside the cell, and that's where the action is in terms of telling what the cell inside what's happening outside. And that part is completely different between the two. So one of the things we're really interested in is what does that difference mean for how each version of the protein can tell um, the cell what to do when there's contact with another cell. They're not mutations. They're naturally occurring variations. And as I was saying earlier, it's quite normal. Different genes, all kinds of different genes do this. They make multiple different sets of instructions for making a variety of different proteins. And so that's what's going on here. So it's a normal thing for genes to do, and CRB1 does it as well. It's just a particularly striking thing that somehow the B1 hadn't been noticed for many years. And so that's, we could imagine a different history where we knew about B the whole time and we knew they were both there and important, but that wasn't how it played out.
Okay, another question. Can you please tell us if your protein therapies in the future will potentially restore vision? You mentioned that the VA of mice improved.
Yes, so that is something we hope, but we don't know. So there's a few different ways it could happen. Um, if, as we think, the external limiting membrane is the key site where CRB1 is acting and where you start to have problems if it's not there, um, it's possible that if we put back CRB1, then you can start remaking junctions that were missing and you could actually fix things. And so in that case, that would be the best case scenario where um, you might really completely stop any further cell loss. It's also possible, in a slightly less optimistic scenario, that while you can stop further damage from happening when you put back CRB1, the damage that's happened is not reversible. In which case, now once the photoreceptor cells are gone, you can't replace them. But what the optimistic scenario is saying is that maybe there's damage at the junctions that has not yet killed cells, but you could reverse that, and then you would have a much more effective stopping of any further cell loss. Um, but it's also possible that the damage is not reversible, and in which case, what you would be doing is sort of holding steady at whatever level that you're at the time you get the therapy. So we're going to do tests in our mouse model of that question, like how reversible is this? And that'll be really important for informing what's likely to happen if we were to get this tried out in people.
Okay, we have time for a couple of more questions. And if we don't get to your question, don't worry, we're going to make sure to answer every single one of these. We will follow up with you within the week to get your questions answered. So if one sibling has the mutation of CRB1, will other siblings have the same mutation of RP?
Um, so to have CRB1 disease, you have to inherit from both parents a version of CRB1 that has a mutation because clinically it seems to only cause severe vision problems when both copies of CRB1 are disrupted. So if there was a sibling that inherited only the mutated version from, and then so in parents, we should add, since unless they have vision problems as well, they probably only have one mutated copy, and then they have one non-mutated copy. So you could have siblings that inherit two non-mutated copies from the parents, in which case they would not be carriers. You could have siblings that inherit just one, in which case they'd be a carrier but not affected, and then could also have a sibling that inherit both, and then they would have the disease.
Thank you. Last question for this portion. How long do you think it will be until clinical trials would begin?
That's a great question. There's so many factors that affect that. Some of it is funding. So FFB is really important in allowing us to do the basic science pilot work that we need to do here to have compelling data to go to other groups like companies or perhaps the federal government to say we need a larger amount of money to really push this to clinical trials. And so there's questions about how much the federal government will prioritize work like this going forward, which I think if we care about turning basic research into therapies in vision science, we need to make our voices heard in the political sphere to our representatives about how important that is. And then the other part of it is that we need to convince companies that it's worth funding this kind of work at a level that FFB, it would be another order of magnitude over that to take things to a clinical trial. So the speed at which it happens depends on the interest of companies in pushing it forward, I think, really. And then, of course, there's still uncertainty if it will work. There's different milestones we would need to hit to keep the things moving forward towards clinical trials. So I would say we're at a very exciting early stage. It's not something that's happening overnight.
Okay. Well, thank you very much, Dr. K. I'm going to turn it over now to Angela.
Great. Thank you, Dr. K, and thank you, Joanne, for managing the questions. If you think of additional questions for Dr. K during the second presentation, don't hesitate to add them to the Q&A, and we'll make sure that they get answered after the webinar. But now it's my pleasure to turn it over to our second speaker, Dr. Oleg Alekseev. He's an assistant professor of ophthalmology at Duke University. He dedicates the majority of his time to basic and translational research in the laboratory, but he also sees patients, including those with CRB1 associated disease. He's interested in developing gene-agnostic approaches to extend the longevity of dying photoreceptors in conditions like RP, thereby preserving the vision of affected patients. Dr. Alekseev will provide an overview of CRB1 associated retinal disease and also talk some about that imaging project that he's part of in the PPA. Dr. Alekseev?
Angela, thank you so much for the introduction. It's an absolute pleasure to be speaking to this audience today and to be part of this very exciting research work, spearheaded by the FFB and by Jeremy Kay. So you just heard a fantastic introduction into an overview of all of that work and an introduction to CRB1 and on the more kind of granular scientific level. I'd like to step back a little bit and talk about more clinical features of disease, the types of disease that this mutations in this gene can cause, the experiences that patients can have with due to these mutations, and kind of what we can try to do in the meantime, while we're waiting for hopefully safe and effective gene therapies or some other therapies to come out. So first of all, what you just heard is about how CRB1 is a very critical protein for the retina for photoreceptors in the retina to work properly. many people in this audience are probably very familiar with all of these terms, but some are probably relatively new in their learning. So let me just kind of step back and explain. So in the eye, the front of the eye mainly does with it has to do with focusing light and making sure that the image is formed properly and projected onto your retina in the back. The retina is the part of the eye that is responsible for actually sensing light. And within the retina, we have photoreceptors. And photoreceptors are those cells that are actually doing the seeing, and they're the only thing in the eye that can detect light. Uh, there are two types of photoreceptors, rods and cones, and you're probably aware that rods are mainly in the periphery. They mainly give us peripheral vision and vision in dim light. Whereas cones are sort of the reverse of that. They are in the center. They give us that nice, sharp, visual acuity that goes with central vision. They see in regular lights and they see in color. Um, so because CRB1 is important to both rods and cones, depending on the type of mutation that a specific person has, and depending on other factors that we don't really fully understand at this point, um, CRB1 associated diseases can affect either rods or cones or both. And so what that translates into is this kind of, if you don't delve into it, it seems like a very confusing picture where, why is it that we're talking about one gene, but all these different conditions, are mentioned as a result of it? Well, and this is why, because there are differences in exactly how this protein, the CRB1 protein, is mutated and what else is going on. So, in general, the type of conditions that patients can have with CRB1 mutations, uh, uh, we have, first of all, the classic kind of condition called retinitis pigmentosa, and that's also known as rod cone dystrophy. So that's where rods are affected first and foremost, and cones later on in life. And that has quite a variability of severity and age of onset. So we certainly see patients who are very young, just a few years of age, and they already have signs of these type of conditions. In that case, we call it something like severe early onset retinal dystrophy, or even Leber congenital amaurosis. These are basically all the same type of condition, just with different ages of onset. On the other side of the spectrum, you can have more of a cone rod type of a picture, where cones are getting hit first and rods relatively spared for a longer time. Um, and so that's known as cone dystrophy or cone rod dystrophy. Uh, or also a variety of that is called a maculopathy. So what are the differences from a patient experience standpoint? Basically, if you have one of these RODS first type of diseases like retinitis pigmentosa, um, patients experience difficulty in dim lights, difficulty with peripheral vision, bumping into things, maybe tripping on things. Um, and eventually, the visual field kind of goes from being wide to narrowing down and giving people kind of a tunnel vision experience. With the cone first type of conditions like cone dystrophy or maculopathy, it's the reverse. The peripheral vision is generally fine. These people don't end up bumping into things much. They see fine in the dark, but when it comes to reading, recognizing faces, doing any sort of fine work, that's where the problems are. Um, and then there are some more rare and obscure versions of conditions that I don't want to get into, but I'll just mention one of them. It's CRB1 is particularly associated with something called Coates-like retinitis pigmentosa, and that's basically where some blood vessels can grow inappropriately and leak. So for those rare patients, they may need some additional care from that standpoint. Um, some features of CRB1 disease can be present. We see them, and but they're not a staple of disease. So I'll mention two. One is fluid in the retina, and I'm sure that that sounds familiar to a lot of people in this audience. So these cystic spaces in the retina that are filled with fluid. We don't fully understand where that fluid comes from, but you heard about the significance of CRB1 to the structural integrity of that external limiting membrane. So that's one explanation for where fluid might be coming from. Um, and the other is that we actually see some patients with kind of an inflammatory overlay to their genetic disease. So it's kind of a unrelated or separate but vaguely related kind of an issue. So that's another possibility where that fluid may be coming from. Um, so that's basically the general overview. And again, these conditions are highly variable in their presentation, with anywhere from a young child with poor vision to I have a few patients in my practice who are in their mid-60s and still have near perfect visual acuity, and everything in between. I wanted to now briefly switch to getting back to this the topic that we're discussing here with the project with the imaging work component in which I'm involved. I just wanted to show you an example of an image that we're obtaining from patients with CRB1. And I'll just share my slide here. Mm. All right, so hopefully you can see that. So many people in the audience will be familiar with the regular OCT retinal scan that you see here on the left that I'm showing, which just shows a cross section of the retina, um, and here with the red arrow in the center pointing to the preserved area of photoreceptors. So when we uh, do our kind of advanced imaging approach here with Dr. Sina Fursu that Jeremy has mentioned, we're taking it a step further. We're getting images that are of much higher resolution and are able to give us information about the integrity of that external limiting membrane much at a almost unprecedented level. So to the right, I'm showing here two examples of that type of imaging. So this is not a cross-section anymore. This is basically looking straight at the retina. And you'll notice that there are a lot of little dots or bumps. Every single one of those is an individual photoreceptor. So I hope you can appreciate just how detailed we're able to get here with this cutting edge technology. And so our hope is to be able to capture as many high quality images like this as possible from patients with CRB1 diseases who are who still, they're not in advanced stages of their disease, but early on and still have an appreciable area of spared retina, so we can get a glimpse into a better understanding of how this disease works on a tissue anatomy level. So I'll stop sharing at this point. And this is significant for two main reasons. One is just to improve our understanding of what happens so that we can design a better drug when, as all this work is ongoing that Jeremy explained, this would guide us better to make a safer and more effective drug. And the other part of it is if we know that there are certain anatomic defects that occur in the course of this disease, we can monitor those defects in patients who are undergoing, who are participating in interventional clinical trials. Let's say a new gene therapy is being put forth and patients receive it. Well, this becomes a metric, another way to see, is this effective or not? So very important work. Certainly, we've had some patients who are eager to contribute to this progress, visiting us from other parts of the country to just to undergo these type of imaging studies. So if anyone is interested, we're always very happy to to see you come in and participate in this. Just let us know. Um, and finally, what I'd like to talk about, I think, the question that I typically get from my patients, okay, it's great to hear that there is research going on. It's great to hear about all this progress. It's great to hear about the interest. But right now, we still don't have something approved. What can I do now to protect my vision, to protect my, family members' vision? Um, so what I would say, there are a couple of things here. First of all, I've mentioned that many patients with CRB1 related conditions can have this fluid in their retina. That is something that I typically try to address first. That's a bit of a low-hanging fruit. Fluid in the retina is never a great thing to have for the health and longevity of the retina. So we try to minimize that as much as possible. There are good eye drops that can be used. Many probably have heard of things like dorzolamide and Trusop. So that's what we typically do for that fluid. Occasionally, it can be quite difficult to get rid of it. And so sometimes we even have to escalate treatment to things like steroids. But that's kind of getting into the weeds of it. Um, I also mentioned the inflammatory component. So I want to emphasize that's a minority of patients. I'm not saying that this applies to everyone by any means, but some patients do have a real inflammatory component of disease, and in those cases, they need to undergo a detailed evaluation. And we have used things like injectable steroids and systemic anti-inflammatory medications to help improve and preserve vision there. And typically, that's a kind of multifaceted team approach with ocular inflammation specialists and often rheumatologists. Um, what I do want to mention is because any patient suffering from a genetic retinal degenerative condition has a higher risk of developing cataracts at a relatively early age. So um, what I want to make sure is that my patients are evaluated for any inflammatory components before they undergo cataract surgery, because that's kind of a step where they're exposed to a higher risk of inflammation due to the surgery itself. Um, and then there are lifestyle modifications. This is not something that's a game changer, but while we're waiting for gene therapies to come up, we want to protect that, those photoreceptors that are intact, so that when the gene therapy is here, we have, a recipient for the gene therapy in the back of the eye. So one thing is, something as simple as wearing sunglasses, preferably polarized sunglasses. And why am I recommending that? There is healthy photoreceptors can handle light pretty well. That's what they're designed for. Photoreceptors that are ailing are not so good at handling stress. And so exposure to excessive light, particularly sunlight that's very high in energy, can push them over the edge and shorten their lifespan unnecessarily. Um, Another thing that's in the same vein is avoiding oxidants. Oxidants are also a stressful thing to photoreceptors, but so oxidants are chemicals, and they push the photoreceptors over the edge before they need to go in these conditions. The main source of oxidants in our daily life is basically from diet, from foods that are very rich in energy. So these are sugary foods and fatty foods. This is why we love these foods. Because they have so much. They're an excellent source of energy, but unfortunately not so great for these ailing photoreceptors. So a balanced diet is basically the solution to that. And then one easy, or I guess maybe not so easy, thing is to avoid smoking. And because smoking, in addition to the nicotine, there are hundreds and hundreds of hazardous chemicals that come in smoke. And they, for whatever reason, they're particularly bad for the retina. So when I recommend not smoking to my patients, it's a different, I feel like in general, people have this kind kind of alarm fatigue from doctors telling them not to smoke. Yes, of course, it's bad for your body in general, but in this case, this is a particularly kind of specific recommendation. For patients with CRB1, it's particularly urgent to quit smoking if you're trying to hold on to those photoreceptors. Um, and then kind of along the lines of mitigating these various environmental exposures, what can we do? I talked about what we can avoid, but what can we do? Um, so there are a couple of supplements that I typically consider in CRB1 patients. One of them, well, two of them are lutein and zeaxanthin. Probably many of you have heard of those things. Lutein and zeaxanthin are naturally occurring compounds that are kind of concentrated in that central part of the retina, and they support the retina there. So by taking these supplements, you're basically getting more of that good stuff. Um, Another thing is something called NAC, N-acetylcysteine. It's an antioxidant. It is, an approved medication for other reasons. It's currently going through trials for RP specifically. But there is enough preclinical data to suggest that it may be a good idea to protect photoreceptors from those oxidants that I mentioned. Um, and then finally, another thing to avoid is actually vitamin E supplementation. So, it's been shown in a few studies now repeatedly the vitamin E expedites the progression of retinitis pigmentosa. Um, and I wouldn't worry about getting it from foods. It's very hard to overdo it from dietary sources. But in synthetic kind of man-made sources like a supplement or a multivitamin, um, you can easily overdo it from there. So I typically recommend to my patients avoiding multivitamin supplementation. And if you need some vitamin for medical reasons, it's always possible to get it in isolation. So those are the things that we can think of while waiting for therapies to come up, which hopefully, fingers crossed, it happens soon. Um, and I think this is what I had to share with the audience.
Okay, thank you so much, Dr. Alekseev. So some questions coming in. In addition to yourself, who are some physicians in the US who are researching CRB1 that you would recommend patients see for care?
Yeah, so the strange thing about our field is that it is when you think about how many people there are who are affected by genetic causes of retinal blindness, there are a lot of people like that in the US and in the world. But when you think about every individual gene, there are relatively few because there are hundreds of genes that cause these conditions. And just like we're having this, webinar on CRB1 gene, there are webinars and meetings like this on many, many other genes. So, um, basically the consequence of that is that there are not that many, everyone kind of studies their thing, and there are not that many people who are studying all this one thing. I think the work that Jeremy Kay is leading with this group is probably the most um, kind of revolutionary work that we have for CRB1 in the field right now. So certainly, we at Duke, would welcome any CRB1 patients. My practice partner, who's also a clinician, here at Duke, is Ramiro Maldonado. He's also open to seeing any patients like this. Um, Another IRD specialist, inherited retinal disease specialist, who has a particular interest in CRB1 is Alessandro Iannacone, and he is at the University of North Carolina. Um, and um, and he particularly is interested in the inflammatory overlay of this disease, so he would be a great person to see as well. But really, we have a small community, and if there is any cutting edge research going on, we all know about it. So it's not so important to be in a particular place. Uh, it is more important to be at a good center where you have access and follow with an inherited retinal disease specialist. And if something happens, that physician will loop you in and make sure that you're plugged into whatever needs to be plugged in, like clinical trials.
Okay. Next question. If one participates in a gene agnostic therapy when it is available, will that negate their opportunity to participate in a potential CRB1 treatment in the future?
So that's a good question, and could be even more generalized to, any therapy. If you participate in any trial, will that negate your future trial participation? Um, so the answer depends on what you're participating in. Typically, if we're talking about like what gene agnostic, like you're asking, some gene agnostic treatment modalities are designed to be, on the gene therapy platform and would require subretinal injection with an operation and all that. So, and that may very well exclude you from being a candidate for other trials in the future. Uh, Other modalities may not be so invasive. So something like an oral pill, for example, or maybe an injection into the eye, but not under the retina. Those are less invasive and are very likely to not compromise your chances of participating in future trials. Um, when it comes to so that's about trials, but when it comes to approved therapies, so if there is an FDA approved um, gene therapy at some point, um, I would be very surprised if they would exclude prior clinical study participants, even if they had been through a surgery. Because the reason for exclusion is because trials have to be designed in a very careful way where we're studying the effect of the medication in isolation. But if a medication is approved already, we're not so concerned about that, so basically, if you have the bottom line is if you have some sort of an operation in the course of a clinical trial, it'll probably prevent you from participating in future clinical trials, but most likely not prevent you from getting an approved treatment in the future.
Next question. Are you using AI in your imaging study? If so, how?
Um, at this point, we are not using AI. Um, AI is something that is very widely used, typically for a somewhat different kind of retinal imaging. We use it for other projects at Duke quite extensively. For this project, really without getting into the excruciating details, it's basically of a different nature, and we're really focusing on just being able to visualize really, really fine structures. So we don't need AI to help us understand what we're looking at. We actually need just to be able to look at it and see it at that high level of resolution.
Okay. So this is next question is in reference to the slide that you shared. What was the difference between the top image on the right and the bottom image on the right that you showed? The images were very different.
Yeah, there are different images of the same area, and the difference has to do with how the signal is processed. So, I'm not a biomedical engineer by any means, so I couldn't, I don't know who's asking the question. If you're an engineer and expect some, a very in-depth answer, I cannot provide that. But what I can tell you is it, there is a signal that's obtained from by performing this imaging of the retina, and it's basically we obtain a three-dimensional kind of um, a sample of the tissue and reconstruct it based on that signal, and then that data can be processed to further improve our ability to understand what we're seeing, and make it crispier, better resolution. And so those the two images you're referring to were two different processing modalities that would allow us to just highlight different things depending on what we're looking at.
Okay. The next question, does CRB1 impact PDE6B and or ABCA4 genes?
So I'm not quite sure what that exactly is asking. There are two ways to understand that. Is CRB1, one way to interpret that is CRB1 protein interacting with the ABCA4 protein and PD6 beta protein. So not as far as we know. I'm not aware of any evidence to suggest that those are directly interacting on the kind of scientific level. And then the other way, which I think may be what this person is asking, is if there are if a person has mutations in CRB1 and also mutations in ABCA4 or PD6 beta, what happens in that situation? So it really depends on on the specifics, what mutations you have and how everything looks. But basically, occasionally, it's very rare, but occasionally we have patients who unbelievably have, two or even sometimes three rare genetic conditions at the same time. So if that were to happen, if somebody had a CRB1 related disease and an ABCA4 related disease, typically, basically you have kind of a double dose of disease. It's not a specific interaction between the two, but it's just you know a worsening of the situation because now you have two diseases instead of one.
Thank you. Next question. How is CRB1 associated disease diagnosed?
So it's a good question. The answer is basically very similar to other inherited retinal diseases. So there are a couple of components, and this will sound very familiar to those families who've gone through the diagnostic process. One component is gathering information. So you're kind of understanding what your experience is, what your very early signs of vision issues have been, and how things have evolved. All of that gives us kind of a glimpse into what may be going on. Second component is objective data. So going in and doing extensive evaluation of the retina with imaging and tests like electroretinography and other related tests. And that kind of helps us further narrow it down to understand which narrow group of diseases we're looking at. And then finally, the most definitive way to pinpoint the diagnosis is to do genetic testing. Without genetic testing, some types of CRB1 disease can be diagnosed just by evaluation, but not most. And that genetic testing is the game changer that allows us to say exactly, yes, there is that CRB1 mutation. This is what it is. This is how it looks. And often we need to also test family members to make sure that everything makes sense as far as how these mutations are inherited, and that typically completes the process for most patients for diagnosing.
Okay, thank you. Next question. Are research teams looking for patients with CRB1 disease to do image scanning? Our daughter is 6 years old and we are in California. There seem to be so many different flavors of this disease, and we would be willing to let her retinas be scanned.
Yes, so we are, obviously we're at Duke. We are one of those research teams who are actively looking for patients with CRB1 disease to do a scan. In particular, early stages of disease where you still have a lot of photoreceptors that are intact and we can image them. So if that's something, thank you for your interest, and that sounds very generous of you to be willing to travel all the way from California to here. And if that's something you are interested in, let's get in touch and facilitate that.
Okay. So our next question, with great presentations, we have lots of questions. So is there a correlation to swelling like I seem to have controlled with the placement of steroids in my right eye?
The correlation was between what and swelling?
I'm not sure. Just that's exactly the question. So if the individual
Yeah.
who asked,
So in a couple of things that I can think of here is, is there a correlation between vision and swelling, like visual acuity and swelling? And there can be. So if you have a lot of fluid in the retina, um, it can distort the image and make your visual acuity, by when you read the chart, not as sharp as it otherwise would be. And that is the reason why you have those steroid implants in there to bring down the amount of that swelling and improve the vision. But the other part of it is uh, kind of more long term, thinking about preserving the retina. Inflammation that is out of control is never good. And uh, there's the genetic component, and it does what it does. At this point, we unfortunately cannot influence it. But the good thing is that it's typically relatively slow. The inflammatory component is kind of a wild card, and it can, if it's significant, it can really make the disease go faster. And so, in that sense, yes, there is a correlation. If we're talking about the fluid being also of the inflammatory origin, then the presumption is that there's inflammation in the retina, and in that case, there's a correlation with, most likely, there is a correlation with progression, in which case, that steroid implant that you have also serves the function of slowing things down as far as the course of the CRB1 degeneration.
and next question, as patients wait for treatment, are contact lenses good or bad for patients with RP?
So as far as the impact on RP itself, contact lenses are irrelevant. So you can have contact lenses or not have them. That's not going to affect the course of RP. As far as your visual experience, it really is up to the individual patient and what works for you. I don't have any contraindications to using contact lenses in any inherited retinal diseases, including CRB1.
Okay. And next question, if optic nerve damage from several laser and vitro surgeries with no lens, can the other eye be jeopardized with cataract and possibly retinal degeneration? Can 100% vision be restored? Are there any trials or studies?
That's a bit of a complex question. I'm not sure. It sounds like this person had a kind of a complicated ocular history. Um, I'm not quite sure how to answer the clinical part of it. Um, if there was a mention of a cataract, if there's a cataract and it can be removed safely, then that typically should improve vision. And then there was a question about restoring vision 100%. So
Correct.
uh, I typically, when it comes to inherited retinal diseases, we don't have right now a way to restore vision, even 50%, unfortunately, let alone 100. There are clinical trials and preclinical work that is going on that's very promising for restorative approaches. So, and this would apply to patients who are affected with, the more central type of CRB1 disease, like maculopathies and cone rod dystrophy. There are restorative approaches that aim to bring back something akin to vision, regardless of the etiology of original vision loss. And it's a whole field. We can talk about things like stem cells and optogenetics and artificial retina implants. So those right now, none of them are FDA approved, but hopefully something will be in the coming years. And at that point, they presumably will be able to restore some vision, but I doubt that will be 100%.
And we'll have one more question, and then again, we will follow up with all of your questions within the week. How can, and this several individuals asked this, how can fluid on the retina be treated?
So what I mentioned was our first line usually is using drops, medication eye drops that uh, basically, if you think about the retina, as far as fluid in the retina, as kind of a sink. It has a drain and it has a faucet that's filling it up. So there are medication eye drops that kind of turn off the faucet, and so you allow the fluid to drain, and keep the retina more or less dry without fluid. Those drops are called things like dorzolamide or Trusub. There are other variations of it. Those are probably very familiar names to some people in this audience. And then for the more refractory situations where we're using the drops, we're doing it, but the fluid is not budging or maybe not going away completely. We can add some other drops. There are the kind of medications basically like ibuprofen for your eye kind of medication in a drop form, or we can go in with more with stronger alternatives like steroids. Steroids are delivered in different ways. So you heard a question from a participant who had steroid implants in their eyes, and that's an excellent way to do it. Or we can administer steroid injections kind of around the eye, or actually inside of the eye as well, if necessary. And then finally, for if needed, we can escalate it to systemic therapy as well. That's not, I don't prefer to do that, and it's fortunately not common that we need to do it, but systemic anti-inflammatory medications and immunosuppressant medications are something that we've successfully used in patients with refractory fluid.
All right, great. Well, thank you so much, Dr. Alekseev and Dr. K, for being with us today. If any of you in the audience are interested in learning more about CRB1 associated disease, we at the Foundation have a webpage dedicated to CRB1 with a scientific landscape document. As Joanne said, we'll reach out to answer all of your questions afterwards. Thank you again so much for participating, and I'll turn it back to Joanne just to close us out.
Okay, thanks so much, Angela. Again, on behalf of everyone at the Foundation Fighting Blindness, thank you for joining us today. As we shared in the opening, we had over 300 people registered from all over the world. So thank you so much for spending part of your weekend with us. And lastly, just wanted to say, if you haven't done so already, please take a moment to visit fightingblindness.org/visions 2026. The foundation will be hosting Visions in 2026 in partnership with Retina International, June 10th through the 13th in 2026 in Fort Worth, Texas, where we're expecting over 1,000 attendees. Very eager to learn more about cutting edge research and therapies, take part in networking opportunities, and attend expert-led sessions and workshops. And this concludes our session. So enjoy the rest of your weekend. Thank you very much for being with us today.
Thank you.